In the plants unit, we will take a closer look at how plants grow, from making observations at the macroscopic scale to looking deep inside at the processes happening at the cellular, molecular, and even atomic level. We all know that plants grow, but have you ever asked how plants grow? Where do they get the stuff to build new plant material? Where does the mass of a plant come from? In the plants unit, we will conduct an investigation to help us explain how plants grow. And we will use the 3 questions, including the movement question, the carbon question, and the energy question to help us understand what our evidence is telling us about how plants grow. But how can we measure plant growth? This is a real brain teaser. When we talk about plants growing, we often mean that they grow taller or stronger, or that they have more leaves. But measuring height and strength or counting leaves doesn't really help us think about where the matter comes from for the plant to grow. That is why we use the 3 questions to focus our thinking. Look at the movement question on the 3 questions poster. Where are atoms moving from, and where are atoms going to? Now look over to the connecting atoms to evidence column. It says when materials change mass, atoms are moving. So in order to figure out where atoms are moving, we'll start by measuring the mass change of our plants. This will help us figure out how plants grow. But measuring how a plant changes mass as it grows is challenging. Why? Plants are mostly made up of water. Think about this. If you stand on a scale, then pick up and drink a big glass of water, will your mass increase? Yes, because you added the mass of the water to your body. But did you grow? Drinking the water made you add mass, so you know water molecules were moving from the glass to your body. But we don't really think about that as growing. Growth is not a very scientifically precise term. In Carbon Time, we look at growth as a measure of how living things add matter to build more parts and build larger parts. When we say that a person, a plant, or an animal grows, we don't mean that it is just adding water. So, how do we account for the growth of a plant in a way that separates out the water? To help us think about the problem, let's first consider one of the materials we'll use in the investigation. The clear gel-like material in which our plants will take root starts out as dry crystals. These crystals are pretty neat. They can absorb water. Lots of water. So what do you think? Are the crystals growing? Remember, growth is not a precise term, so you might argue either way. But the crystals did gain mass by absorbing water, meaning atoms that make up water, H2O molecules, were moving. So then, how can we measure the mass of the crystals? We can mass them on a scale. First, find the mass of the empty container, or tare, then add the crystals. Next, we can subtract the tare from the total mass to find the mass of just the crystals. This number is the dry mass of the crystals. Now, let's find out what happens to the mass when we hydrate or add water to the crystals to make a mixture. The mass of the crystals is now much more. H2O or water molecules have added mass, wet mass, to the mixture. Once we know both the total mass of a mixture and its dry mass, we can calculate the percent dry mass. This percentage can be used to estimate dry mass in the future. Knowing the dry mass percentage, we can estimate the solid mass that is in any amount of gel mixture. We can now figure out dry mass of a mixture when we add water to a dried substance. But what if we start with a mixture? Let's look at another example. To help our plants grow, we'll use a little bit of nutrient mixture. How can we find out how much dry mass is in this mixture? Let's mass it. Next, we can evaporate out the water and measure the mass of what's left. Again, we can calculate the percent dry mass, just like in the crystal and gel example. A quick note, when we use the nutrient solution to hydrate the plant gel, we'll dilute it with much more water, so the final percent dry mass will be much less than shown here. This example shows us that another way to measure dry mass in a mixture is to dehydrate it, or take the water out of the mixture. Let's think about our growing plants again. Now we know how to separate the water molecules out from a mixture, which will help us measure growth in our plants. But if we aren't measuring the water molecules in plants, what are we measuring? Let's look back at the 3 questions. In Carbon Time, we're interested in tracing carbon atoms to explain how plants transform matter and energy as they grow. Molecules containing carbon make up the matter that the plants gain to make bigger or more leaves, stems, and roots. We will measure or calculate the dry mass of our plants both before and after they grow, keeping track of the dry mass in the seeds and the gel when we plant, how much dry mass is in the nutrient mixture, and how much dry mass is in the plants and the gel after the plants have grown. This will tell us how much of a plant's mass gain is from an increase in molecules containing carbon. Finally, the 3 questions are about more than just carbon and water. As we collect data on our plants, we will be collecting evidence to answer these other questions as well. So, let's get started.